Okay, so are there any questions from last lecture? Okay, I noticed that 400 and some odd students in this lecture hall, nobody did the time work, but that's fine. I don't know what I was expecting, but so what I want to do today is finish up the last lecture and then begin the next. If you remember correctly, last time we were talking about selection and I was talking about experimental evolution. I gave one example of experimental evolution in viruses, the X174 example. That was an example of adaptation to high temperature for viruses. I want to give you another example of experimental evolution. I kind of almost hesitate to give this lecture to most of the people in this room because you guys are all in your early 20s and probably have no appreciation for what it means to get older. Basically, you're at the top of the game right now physically, and probably mentally too. But from here on out, trust me, it's all downhill. All right, it's all downhill. You've got a lot to look forward to, but you might as well enjoy it while it lasts. So the problem, of course, is well, humans senesce. That they age as you get older. Things fall apart, believe me. And it turns out it's not unique to humans. You all know that you have pets that have probably died in your lifetime, and it's not just that they die, but they get old, right? But decrepit before they die. And this is a fairly universal observation. So in vertebrates, old age is associated with a higher incidence of cancers, for example, right? Insects, as an example, also senesce. They don't really get cancers anymore frequently, but an old fly, for example, looks like an old fly, has tattered wings. Its physiology is much poorer than it was when it was a young fly, right? So animals senesce. And not only that, senescence, the rate at which organisms age or senesce, evolves. So you look at a mouse. How long does a mouse live? A couple years. How long does a human live? Maybe 80, right? Maybe the top age I think any human ever recorded is maybe 122. Right? That's probably the upper ceiling on how long a human can live, somewhere around there. So between a mouse living maybe three years and a human living on average at least now 80 years, there's been a lot of evolution at the rate at which these organisms age. Even if you look at our closest relatives, the chimpanzees, they don't have a lifespan that matches that of humans. They live in even the most benign conditions, maybe 50 or 60 years, and then you have a very old chimp that dies. Okay? So I want to talk about the evolutionary explanation for senescence, and then a really cute experiment that was done, okay, to test this hypothesis. So to understand that evolutionary explanation for senescence, I want you to do a thought experiment. Right? The thought experiment is to imagine an organism, a species that does not senesce. That is to say, as they progress, as the organism gets older and older, they're as fit as you guys are, right? They're like 20-year-olds forever. Okay? So there's no aging, right? But that doesn't mean you don't die, right? Just because you don't age doesn't mean you don't die. You still have a chance that if we imagine that this is this idealized population of gazelle that doesn't age, it still doesn't mean you don't get chased down by a lion and eaten. And right? That would be an example of not aging, but real gazelle age. But our imaginary gazelle don't age, but they still die. If you're a human, if you imagine a human population or imaginary human species that doesn't age, still get hit by bus, right? Still have all the accidents that can still befall upon humans still might occur, and you can still die. So we would imagine we have this idealized population. I'm just going to label the cohorts: year 1, year 2, year 3, and so forth. So we imagine that we have our imaginary population of in the species that doesn't age. We have a bunch of individuals born on some year. Right? So here they are. Here they are, represented by the frequency of the number of individuals. It doesn't really matter. And if we imagine this case, this ageless species, let's say 5% of them die every year. Well, that means the next year you're going to have fewer of them, 5% fewer. Then you'll have 5% of these guys will make it. So as we go through time, the cohort, the number of individuals from this original cohort that are still alive, say 5 years from now, will be many fewer than what were alive to begin with. So if the rate, I put this in my notes just so I did the calculations, but how many individuals, so if you have a death rate of 5% a year, that's to say 5% of the organisms drop dead, maybe they get eaten by lions. At 5 years, 77% of the original cohort will be alive. At 50 years, 7% of this cohort will be still alive. Right? So that's not, I'm just saying that this is our imaginary non-senescing species. Right? As they get older, fewer and fewer from this cohort will be around. So we can still think about this cohort going through time. These are the individuals that are young, and these are the old individuals. You still have young and old individuals, right? But they're just the old individuals look just like the young individuals in terms of their vigor and their fitness. Okay. Right, so that's the first thing I want to point out. So what is the evolutionary explanation for aging? So it involves the thought experiment of what happens to mutations that occur in old and young individuals. So let's imagine a mutation that's beneficial in both old and young individuals. That's our first imaginary example we're going to look at. This is a mutation that's unambiguously beneficial, right? It's good in both the young and the old. And natural selection says that this mutation will spread through the population, right? On average, mutations like that will spread through the population. Okay, and of course, if it's deleterious in young and old individuals, through purifying selection, that will be removed. Once again, these are the cases that are pretty easy to understand. Unambiguously deleterious mutation, that is to say, it affects the young and the old in the same way. It will be removed by natural selection through the process we call purifying selection. Now, things become a little bit more complicated when we imagine a mutation that might be beneficial for young individuals and deleterious for old individuals. The question is, will that mutation spread through the population, or will it be removed by purifying selection? Okay, it's not clear. Well, it turns out that there are scenarios where you have a mutation that's beneficial in young individuals and deleterious in old individuals in which that mutation will spread. And the reason why it will spread is that beneficial mutation acts on many more individuals. There's many more individuals around that are young than that are old. So this deleterious mutation doesn't affect that many individuals. Natural selection can't grab hold of it and change its frequency as readily. Okay? And then this type of mutation, deleterious and positive, will probably, on average, be removed. Now, the details of whether it be removed or spread really depend on the fitness effects of the deleterious and positive mutation, but I'm just making this a very simplified example. So mutations that are deleterious in young but beneficial in the old will be removed in this scenario because there's so many more individuals, again, that are experiencing the deleterious effect. Right? So basically what I'm saying is that natural selection does not see old individuals as readily as it sees young individuals because there's many more young individuals around in the population on which natural selection can act. Right? So that is the evolutionary kind of a simplified version of mathematical models that describe how senescence might evolve. The main point here is that you have mutations fixing in the population that have beneficial effects in the young but are deleterious in the old. So one of the explanations for senescence is that you're seeing the effect of a lot of these deleterious mutations arise. Right? So all these diseases are associated with old age. The idea is that many of them are just a manifestation of these deleterious mutations that were fixed because there was some positive effect in the youngsters. Okay? So how might we test this? Well, there's this interesting experiment that Michael Rose, who's a professor at UC Irvine, he did. This is an experiment that's now probably about 20 years old. And what he did is he worked with the Drosophila melanogaster, the fruit fly. And he did an experiment as follows. So normally when you rear flies in a laboratory, it's kind of a smelly proposition, but you have these vials that are about this big. In the bottom of the vial, you put usually a substance that the fly maggots like to eat. It's usually a banana or some sort of meal, I'm not exactly certain what it's made of, but I know it smells. And you have a stopper here that is permeable to air. And you have your little flies in here. There's flies flying around. If you have males and females in the tube, you can imagine what happens. They lay eggs on the surface here, and then these eggs hatch into maggots, and they crawl around, and eventually they pupate and they hatch into adults. And what you do is one of the jobs in a laboratory that works for fruit flies is you need to keep transferring the flies to fresh vials because, of course, they eat up their food and they file their vials, right? So you want to keep transferring them to fresh vials. So what Michael Rose did is this. He had a vial of flies. They laid their eggs. He took out all the flies. All the adult flies were removed from the vial, and he waited for the maggots to pupate, and he had little flies, right? Adult little flies, young flies. He immediately separated the males from the females, right? And put the males in one jar, one tube, and the females into another tube. Okay? So now we have two tubes, males and females. He didn't let the males and females combine. He didn't combine them into one jar until the flies got to a certain age. So he waited, say, a couple weeks. So I should mention, by the way, that normally flies live about 3 weeks, right? 30 days maybe. And a 30-day-old fly looks like an old fly, right? It's not a vigorous fly. It looks like a senescent fly. It's nothing compared to a young, vigorous 1-week-old fly. Right, so he basically combined these flies at later and later dates. So the flies that mated were the ones that successfully lived to a certain age, say 2 weeks or 3 weeks or 4 weeks or 5 weeks. He progressively set the bar higher and higher, letting the flies only come into contact after a longer and longer amount of time. So basically what he's doing is he's forcing, in order to mate and reproduce, you have to be a fairly old fly, right? And then as time went on, he raised the bar and you had to become a still older fly to mate and reproduce. What he was doing in this experiment is making these mutations, these deleterious mutations that occurred late in life, important again, right? If you have lots of mutations like this that occur late in life, you're not going to be, and you're a fly that bears these mutations in this file, you're not likely to survive to reproductive age, right? And so purifying selection can now act to remove these deleterious mutations. So after 12 generations rather, of passaging flies in this manner, he went from flies that had a maximum lifespan of 30 days to flies that had a maximum lifespan of 40 days. That's after 12 passages like this. He's continued these experiments, and he's done all sorts of things with them since, but he's continued selecting for long life in these flies, and he now has flies that can live almost half a year. Remember that they started off with living about a month, which is pretty cool, right? That'd be like a human living, what? Well, a long time, right? I can't do the math right now, but it's a long, long time. I guess it depends on what you think the maximum lifespan of a human is. But it's 500 or 600 years. It's a long time, which would be pretty cool. So he kind of jokingly calls this kind of the career woman experiment in flies, because a lot of career women are actually putting off having children later and later in life. Right? Anyway, so this is an example of natural selection, or selection used to explicitly test this hypothesis. So the idea being that you're removing, through purifying selection, these late-acting mutations, and that's what's allowing these flies over time to live longer. And then there's a couple other things you can test as well. So for instance, if this is the case, if senescence in these flies is caused by mutations are beneficial to the young but deleterious in the old, well, so what types of beneficial mutations might you have in the young? Well, maybe a beneficial mutation would be something like I'm going to divert more of my resources to my eggs, right? And by more of your energy budget. And by doing that, you sacrifice something later in life. This would just be, there's all sorts of examples or scenarios we might come up with. The details, of course, would vary from organism to organism. But if that's the case, by removing this mutation that has this beneficial / deleterious effect, depending on age, what you should see then is in these flies that are living a long time, there should be some trade-off they've made, right? And so what you see in these flies is that even though they're incredibly vigorous and they live a long time, they don't have as many offspring. So if you imagine selection is all about having lots and lots of kids early in life as fast as you can, and you're a fly where there's all sorts of predators that can pick you off, so your death rate per year is probably much higher than 5%, then you might expect that any mutation that allows you to have lots and lots of young really quickly would be beneficial. Now that all that's been removed in this artificial laboratory condition, and so now you see these flies, because this mutation has been removed, they don't also have lots and lots of the beneficial the effects that it had in young flies. So that's a pretty cool example, I think. All right, so I'm going to change now to an example of natural selection in the wild. I need, there we go. And here's the challenge I was faced with. So one of the nice things about teaching this course is I can give you the very, very best from evolutionary biology. I can choose from an entire huge literature that involves tens of thousands of papers, and if I want to talk about natural selection in the wild, I can choose whatever I want, but I can choose the very best examples. And I decided in this case to not only choose a very good example, but one that you guys might be able to identify with. This is Professor Hoekstra. Hopi Hoekstra is her name. This is her in the field. She's a mammologist. She's currently at Harvard University. She's a professor there. But she started off at UC Berkeley. She graduated in the early 90s. She took Y1B, she tells me around 1993, which doesn't seem that long ago to me, although I know you guys were youngsters then. So here she is in the 1991. She was a volleyball player here. She's in the bottom left. So I figured I know Hope because she and I were both at UC San Diego before I fled to Berkeley and she fled to Harvard. Here's where she lived. She lived in a sorority on campus, Delta, Delta, Delta. I delve further. Here she is at a party. She's the third from the right. You notice that I chose this one because you can't really tell what the girl on the second woman on the left there is drinking. Sure, it's just soda. And recently she's just had a baby. Everything's going really well for her. But what she studied for her postdoc is the evolution of what's called cryptic coloration. So here's a very famous example involving cryptic coloration in moths, which I'm sure you probably saw in your textbook in high school. This is a fine example even now, although the system's much more complicated than people originally thought. She's a mammologist, so she studies cryptic coloration in mice, right? Mice being mammals. And here's just an example. A lot is known about the genes that underlie co-coloration in mice, especially house mouse, because the mouse is a laboratory workhorse. That is to say, lots and lots of experiments are done on mice, and then there are lots and lots of mutations that affect co-coloration and pattern even in mice. So as an example, here's a bunch of different mutations, genes that are involved in co-coloration. You don't need to remember any of this in case you're wondering. Don't study the list of genes, but there's a lot of genes that affect co-color and pattern in mice. And here's some examples of the different types of colors you can get in mice. All sorts of things, right? And some of these genes are actually shared from humans to mice. Here's a mutation called the piebald mutation. It has a little patch of white hair. There's a piebald mouse. There's a piebald professional basketball player. Okay? It doesn't appear to have any fitness consequences. It's a neutral mutation. Here's another type of mutation that affects co-coloration that you find in all sorts of organisms. There's a number of ways you can become albino. This is an example of a deleterious mutation. Individuals that have their albino have all sorts of fitness, there's fitness effects on these people. There's a reason why we have melanin in our skin. But the important point here is that this is also a mutation that it's the same type of pathway that works in many different species. Okay. Now, the example she worked with was rock pocket mice. She started this work when she became what's called a postdoc. This is a short period of time between the time when you graduate with your PhD and the time you get a real job as a professor, say, somewhere. Not that this is a real job, but we'll call it one for argument's sake. So it's called a postdoctoral period. It's actually usually a very nice time of your life because you don't have any responsibilities except to do research, right? I look back at my postdoc days with envy now, fond memories. But she worked with a fellow at the University of Arizona by the name of Michael Nachman. Michael Nachman is also a UC Berkeley grad. He graduated as undergraduate here in 1983, which is even long ago. It's even old by my standards. but he's now one of the world's leading mammalogists, and he and Popey studied co-coloration in rock pocket mice that live in the deserts in the Southwest. So I don't know how many of you have driven along, say, Highway 8 in Arizona, right, and also New Mexico, but as you drive along the desert, which is a long drive, so you have lots of time to sort of look at the scenery, most of the time you see the environment looks more or less like this, where you have this sort of granitic substrate, like sands, right? But occasionally, you'll drive across these areas which are recent lava flows, recent meaning a couple million years old to a couple 100,000 years old, okay? So here's an example of a lava flow area where, first of all, notice that the rock is basaltic and dark. The substrate is dark. This is a particular area called the Pinacati lava. It's a flow that I can't remember exactly how long ago it occurred, but within about 1 million years ago. There's an example of the mice, the same species that lives on in this area, but you'll note that when you look out in the granitic parts, the light sand parts, the mice tend to have the normal ancestral coat color pelage, sort of this yellowish tan color. But if you look on the lava flows, you'll find mice that have these dark coats. Same species, you can take them in the lab, made them, and they produce little mice, no problem. So they're the same species. Now, we'll get into the fitness effects of why this might be the case, but the obvious argument here is that natural selection might be acting to favor individuals that match the substrate, right? So if you happen to be a mouse living on the lava and you have a light coat like that, maybe you stand out more to predators such as owls and you get eaten at a high rate. Well, she actually tested that particular statement, but that's an obvious explanation that we're going to use as our working hypothesis here. She was also interested in the specific mutations, which we'll get to a bit. But here's an example of some of these different lava flows. Okay, so the Pinacati's lava flow is 1.7 million years old. But you can see that there's these other lava flows that occur, some of which are quite young, and the Pinacati's is the oldest. And what this graph is showing you is that on the lava flow, most of the mice you find have the dark coat, if you go just adjacent to the lava flow, that is to say, you walk off the lava flow into the granitic sands, you find mostly light-colored mice, right? And similarly, in these other lava flows, you find the same pattern: dark on the rocks, on the lava flow, light mice on the normal desert sands, and so forth. So what is going on here? And here's just examples of the coat colors you find on the lava versus off the lava. This is how usually the mice live in the museums here. All right, so what did Popey do? So the first thing she did is she realized that, when she looked in detail at the hairs of the dark-colored mice, the hairs were reminiscent of a mutation that occurs in the house mouse, right? The mouse that people use in laboratories. And so her first thought was maybe the same gene is responsible in the rock pocket mice as in the mouse, the laboratory mouse, right? And so she went in, she sequenced the particular gene. The gene was called MC1R, for whatever that's worth. And the approach she was going to look for is going to use it. She's going to ask, do we see any mutations that are always associated with the dark phenotype? That's what she's going to be looking for. So she's basically saying, when we do the sequencing around these genes, are there mutations that are always associated with being a dark mouse? Okay. So this is the gene once again that she studied, MC1R. Basically, MC1R is a transmembrane protein, and it's a signaling protein. Basically, you have something latches on to the protein from the outside, and it transmits that signal onto the inside of the cell. Right? So in this case, if MSH, melanocyte-stimulating hormone, latches onto the protein, cyclic AMP is activated, and I believe you get the light mouse, even though the arrows make it look like that's not the case. A Goody is a repressor. It's a protein that represses MC1R, and you get the dark mice out in that case. So she's wondering, so you can see that there's a number of different places here where we might expect mutation. It could be in MSH, it could be in a Goody, or it could be in MC1R. Some of the mutations in any of those genes might affect this pathway. So this is, the answer, I'll cut the chase, the answer is it's MC1R. Okay? And this is the raw data she collected. This is, what we see here is not the whole sequence that she collected, but only those positions in the DNA sequences around MC1R where she found any differences. Okay? And the point here is that there are 4 mutations that are always associated with being dark. Okay? 4 mutations that are always associated with being dark. And you can see that there's some individuals here at the top, which we're going to call having 4 of those mutations the big D allele. If you're homozygous for big D, you're dark. If you're heterozygous and have the D allele, you're also dark. Right? So it's kind of a dominant mutation. If you're homozygous for the little D allele, that is to say you don't have any copies of those, you don't have any of those 4 mutations, then you're light. So you find this complete association. There's some other experiments you could have done that would have nailed it down, but this is so clear that you don't really need to do those additional experiments. So this is just to summarize the results. You have, if you have 2 chromosomes, both which bear those 4 mutations, you're dark. If you have one of the chromosomes with the 4 mutations, you're dark. If you don't have neither of your chromosomes has those 4 mutations, you're white. So first thing is she discovered the mutations that are responsible for dark co-coloration in the Pinacati's mice, ones that live on the Pinacati's. And here are where those mutations occur in MC1R. They're all replacement changes. So that's to say they're mutations that cause an amino acid change, and the change in the DNA sequence that causes an amino acid replacement change. Here's the 4 amino acids that are different in MC1R. And note that these mutations are all on either the outside or on the outside and the inside of the mutation of the gene. That is to say they're on the part that faces outside and the part that faces into the cell, which is suggested at least. So here is, so that's first, like I said, she discovered the mutation. What's a little bit more difficult is to ascertain the fitness effects of these mutations on the mice. And that's because it's difficult to get approval to do the obvious experiments. At least now it's difficult to get approval to do the obvious experiments. What would you experiment? Well, put a bunch of mice on light sand and release an owl would be a good example, right? Or put a mixture of light and dark mice on light sand to see which ones are picked off more frequently. So that would be the obvious experiment. You can't do it now, but you could do it in the past, right? And there's a famous experiment by Geis in 1947. He didn't look at predation in the rock pocket mouse that would be directly applicable to what Popey and Michael Nachman were doing, but he studied deer mice. And it was a very simple experiment. He took a big barn. On the floor of the barn, he made like a little arena where he divided the barn in half. On one half, he put light substrate, and the other half, he put more of the salt dark substrate. And he released mice of light and dark co-colors onto the floor, and he put a couple barn owls in the barn, right? And he just looked at the frequency, how rapidly these mice were picked off. And the result was that if you were a mouse that was mismatched, that is to say you had a dark coat, but you were on a light substrate or vice versa, you got eaten at a higher rate. Okay? And Popey has done a similar experiment with clay models. So what she and her graduate students will do is they'll have like a little craft session where they make a bunch of clay mice, soft clay mice, and then they'll paint them, right, dark or light, and put them out in the evening, and the next morning they'll come back and they'll look for evidence of predation. That is to say, little clay models that have little scratch or bite marks on them, right? And you'll find that you have much more evidence of predation in the clay models that are mismatched. Those are the types of experiments you don't need approval to do because they're on clay mice. All right, so and one other thing we can look at is we can look at the distribution of that of those 4 mutations across the desert. So what she did is she took what's called a transect. She just laid out a line and she collected mice along the transect. Okay? And then she looked at the frequency of those 4 mutations in the light sands versus the lava flow versus the light sands again. And the point here being that for those 4 mutations that are responsible for the coat coloration, you see them at high frequency in the rocks and in the lava flows, but not in the sand.